Good morning, good afternoon, and good evening, and welcome to the Bioinformatics and Silico Drug Discovery track on the BioData World Congress 2020. Just a few housekeeping notes before we get started. I'd like to encourage you to ask any questions in the box to the right-hand side of the screen, and through there you'll be able to get a response from the speakers presenting today. So, without further ado, our first speaker of the day is Yuriy Gankin, who's the CSO of Quantori. Yuriy today will be discussing multi-reference poly-conformational computational platform for De-Novo design, optimization, and repositioning of pharmaceutical compounds. Yuriy, the stage is now yours.
Good afternoon. I'm Yuriy Gankin. I'm a co-founder and CSO of Quantori. We develop custom solutions for biopharma and life science technology companies, and we're focusing on data engineering, data science, modeling, and infrastructure design and architecture. Today, I will briefly describe our approach and technologies for computational drug design and repurposing. Typically, scientists seek molecules with selectivity for one target combined with a low affinity to an anti-target or a couple of anti-targets. So a good example of a very successful program or programs which yielded amazing drugs such as Prozac, Paxil, Zoloft, Lexapro, and many others is selective serotonin reuptake inhibitors. So they're usually optimized for 5-HTT encoded by SLC6A4. And people consider certain anti-targets such as 5-HT2B or hERG to make sure that adverse effects such as cognitive impairment, cardiac toxicity, and others are addressed. However, there are many more anti-targets which should be considered and which are responsible for important side effects. And there are notable side effects due to multiple unintended or just, frankly, ignored targets. So the question is, could we hit multiple relevant targets or optimize against multiple reference compounds if target information is not available using the same molecule? Let's use COVID-19 as an example. A good target candidate is M protease. Usually it's used as a main target. M protease is essential for activation of RNA-dependent RNA polymerase, which is virus specific, very attractive target. Then there are other targets, of course, such as human ACE2, which is responsible for binding to the host membrane. We could also add RNA-dependent RNA polymerase as a target, because its inhibition would, of course, lead to termination of virus RNA synthesis. And then it would be attractive to look at additional functional effects, such as increasing of pH, for example, in endosomes to inhibit viral RNA release to cytoplasm. This is a hypothesized effect of chloroquine, for example. And then, of course, we would need to consider anti-targets, such as sodium-potassium channels or CYP2D6, to avoid adverse effects similar to, again, chloroquine or hydrochloroquine adverse effects. So multiple relevant targets could be hit theoretically by different conformations of the same molecule. Of course, in real time, the most molecules exist in multiple conformations or shapes. Those shapes are based on the environmental conditions around the molecule and other factors. In particular, each 3D shape of a molecule dictates its biological activity and enables the molecule to fit into a binding pocket of a protein or bind to another target. Often, distinctly different chemical compounds that have similar shapes and thus similar charge distributions along the molecular surface. So different chemical compounds may act in a similar way. The way scientists see a compound is different from the way enzymes see a compound. So structurally, visually, I would say different 2D structures can have similar 3D shapes and bind to the same target. Therefore, we think it's beneficial to compare the shapes and surface distribution charges for target query and reference compounds, not just on a compound base, but on a conformer by conformer base. We called our approach multi-ref 3D for lack of better name or abbreviation. It's an alignment-free, multi-objective fingerprint-based optimization protocol. So we don't use alignment as it is not universal, it's computationally extensive, and significantly different molecules are hard to align and use the method to identify targets which may look differently but have similar effect on a particular target. So our goal is to maximize 3D overlap of a query molecule, but not just the molecule itself, but the whole conformational ensemble, which is the whole range of conformers which molecule may assume. So we'd like to maximize the overlap of that conformational space with the conformational ensembles of multiple reference ligands. So I will double click on that a little later, but the method is using fingerprints. It's rapid and accurate and universal. So we can compare different molecules using fingerprints. It's applicable to desired search universe, and we're interested in billions of virtual molecules. And if we consider up to 100 conformers per molecule, our search space may grow to hundreds of billions of shapes. So the idea here is that we treat each conformation as an independent entity, which is characterized by a 3D fingerprint. Fingerprints describe 3D shape and distribution of electrostatic charge across the molecular surface. So we generate those fingerprints for each query molecule conformer, not just for each molecule, but for each molecular conformer. And we also generate those fingerprints for each conformer of a reference compound. A reference compound is a compound which shows some efficacy of the compound which would like to mimic in some way or form or shape. So fingerprints for each of the query molecule conformers are individually scored by Euclidean distance as a similarity measure with respect to each conformer of a reference compound. It's computationally very simple and allows us to work with huge libraries of virtual compounds. So we define an objective function with a goal to maximize sum of individual objective functions across all reference compounds of interest. So the idea here is that we would generate maximum reasonable number of conformers for each of the reference molecules, molecules which have shown activity against our target, in case we don't have a target information, for example, and then generate conformers and calculate the fingerprints for all our query compounds. Q stands for query here and R for reference, and then maximize W all for each pairing reference compounds with our query compounds. So similarity is calculated in a relatively straightforward way. The fingerprints for each of the query molecule conformers are individually scored by Euclidean distance, as you can see here. We use it as a similarity measure with respect to each conformer of the reference compound. So, so basically comparing again shapes and charges for each conformer of our query compounds, or virtual compounds, screening compounds, with those of attractive compounds from, say, clinical trials. I think it would be much more clear if we look at a specific example shown in this table. The query compounds from a virtual library are shown in the first column, virtual compounds, and they are sorted by their total overlap score which is shown in the second column, titled Sum. The values in other columns correspond to the sums of the overlap scores of the conformers for the corresponding reference compounds. So we can see here that the top compound, number 1, has a total score of 354 because it resembles, based on fingerprints for the conformer ensemble, they resemble chloroquine, remdesivir, favipiravir, and other compounds. And so it has a high score. Also note that the total score should not be the deciding factor for the choice of a compound. It's important to look at individual scores. We need to take them into consideration. For example, some conformers for compound 14 here, sorry, it's not shown here. Some compounds, such as 11, doesn't have overlap with chloroquine, and compound number 12 doesn't have an overlap with remdesivir, etc. So when we consider the compounds and prioritize them for in vitro screening, for example, we need to take into account overlaps, and we would generally exclude compounds which have 0 overlaps with some of our target molecules. For example, in this case, we would not recommend compound 5, 11, 12, or 13. Obviously, these are used just as an example. In reality, we can filter those compounds during the modeling, and we can filter compounds which have 0 overlap with at least one reference, and use our own ranking functions depending on a particular program or a problem at hand. So, so this is more for illustration purposes. So, so based on this analysis, we would select virtual compounds and rank them for further testing. Here's an example of compounds which we identified this way, selected from a large virtual library. There's the top hits. Note that those are very flexible compounds due to their sulfanyl or, ether bridge on the left, or sulfanyl bridge on the right. So the ring substructures can rotate, which ensures the ability of those molecules to accommodate different targets, and indirectly confirms our hypothesis and the approach idea. And then, of course, we can conduct further structure optimization using De-Novo structure generation or optimization methods. There are many of them, and we can select an optimal structure from a screened virtual library, as I showed in my previous slide. Or we could perform in silico modifications using a set of medchem transformation rules or bioisosteric replacements. There are references here. We could score each modified molecule and select ones which score higher, going back to the table, similar to table above. And I can repeat those steps 2 and 3 until convergence, and select and re-optimize structures until we're satisfied with the results, and then conduct a wet lab cycle to verify their in vitro efficacy. Here is a quick example of multi-conformer optimization, optimized chloroquine, based on the approach shown in the previous slide. So to make the approach which I described efficient, we've developed a set of tools, engines, and call them Quantori Discovery Platform, which is based on integrated informatics engine, which unifies content and informatics services. We've combined content from most relevant public sources, and we created a system which allows us to plug in private proprietary data sources if clients are interested in adding their data sources to the mix. We developed universal queryable format, and this integrated source links compounds with targets through sources such as Campbell, PubChem, DrugBank, PDB, and others. and targets with diseases through open targets, therapeutic targets database, and other well-known data sources. And of course, each entity, each object, such as compounds, targets, and drugs, also have data sources, specific data sources associated with them, such as Unichem or Uniprot. And we've developed common ontologies and control vocabularies to cross-index all those sources and have easy access to the reference compounds as well as virtual compounds. Here we use real library, which contains 1.4 billion compounds, target libraries from enamine and other sources, as well as zinc, and any virtual library could be plugged in to the system. So all the entities are connected, and compound can be directly connected to a disease or phenotype through clinicaltrials.gov links or open targets. So a pretty efficient and well-integrated system, which we can use then in conjunction with our computational and bioinformatics tools and engines and build custom workflows depending on the program or task. So this integrated informatics basis or engine allows us to enter a program from multiple entry points and use different ways to select reference compounds. We can start with a disease and select interesting compounds using clinical trials, open targets, or Campbell. Or we can start with a target. We can look at PDB and extract list of co-crystallized ligands as reference compounds, or from PubChem and extract list of ligands, or we can go directly to virtual libraries and work with real zinc, Unichem, drug bank, or proprietary libraries from pharma and biopharma companies. And then we can use the platform for next generation drug repurposing or novel compound, De-Novo compound design using various approaches such as variational autoencoders or generative adversarial networks. But the most important point of the slide is no matter how we enter the platform and select reference compounds, the heart of the platform is multi-target poly-conformational optimization engine, which allows us to create conform space, calculate 3D fingerprints based on various algorithms and approaches, and then rank the compounds based on their resemblance to a conformer space of all selected reference compounds. So what are the next steps? We've integrated our platform with Quantori's high-performance docking platform to allow for further refinement and verification of candidates in silica. We have an efficient reverse and direct docking platform, which is cloud-based, scales linearly with respect to the size of the problem, and has been shown with several case studies to be efficient computationally and accurate. So it's a relatively precise and rapid way to do further compound ranking, verification, and assessment. And then we also, as we speak, we're adding cross-domain QSAR and machine learning capabilities to enhance De-Novo design capabilities and compound repurposing as well. So also we're working on validation of the system and the structures which we have identified. We plan to open source non-proprietary parts of the platform, and we're also applying our tools to build computational drug discovery workflows for private companies. And we have identified and hope to work with wet lab partners on the COVID project to see if our COVID compounds indeed efficacious and have low toxicity. With that, I would like to finish. Here is my contact information. I would like to thank you very much for your attention and be happy to take any questions now or via email, phone number. I will be available also to one-on-one meetings throughout the conference. So thanks again, and I'm looking forward to talking to you.